Hello, I'm Pietrzyk from the Fraunhofer Development Center X-ray Technology, EZRT in Fürth, Germany. Alongside X-ray technology, we also specialize in developing optical 3D sensor technology and complete measurement systems for various applications. For over 15 years, we've also been active in agriculture, currently focusing on 3D plant phenotyping. In 2021, we initiated the 45G project to develop a digital twin for food cultivation, collaborating with the Friedrich Alexander University, the Weinstefan Triesdorf University, and the Food Information Center in Forchheim. In this project, we used a drone equipped with an RGB camera to fly over the orchard, capturing images of each individual tree from various angles, allowing us to create a 3D model of the tree and analyze it using algorithms for trait extraction. Our initial approach for 3D reconstruction was to use conventional multi-view stereo. This allowed us to collect data throughout the entire season, to capture temporal development and derive information about tree conditions. For example, we were able to monitor tree health or yield, as well as information required for irrigation and fertilizer use. However, multi-view stereo is computationally intensive. Hence, we are using a new approach with neural radiance fields, or NeRF in short. NeRF is a method based on artificial intelligence that can learn 3D scenes from images and associated poses, rendering the object in 3 dimensions or from any arbitrary pose. As seen in this video, we can generate views from poses that were not taken during the drone flight. The capabilities of NeRF extend well beyond 3D reconstruction. With the method FruitNeRF, we can additionally transfer semantic information from 2D images into the 3D representation. For example, we use this for fruit counting, but the method has generalized potential to segment plants into branches, leaves, or other components relatively easily and accurately. We further utilize NeRF for indoor 3D phenotyping by rotating plants on a turntable in our climate chamber at the EZRT. We integrated a sensor system with multiple RGB cameras to capture images of plants, such as this cannabis plant or this wheat plant, from various perspectives. And so far we have achieved quite impressive results. For example, in this point cloud generated from the NeRF model, we can view a wheat plant and its fine structures on the ears in great detail, but also on the rendered video of the NeRF model, we obtain detailed information between the individual plants, even in complex settings, such as densely planted wheat. And while we cannot measure traits directly from the NeRF model yet, we easily generate 3D point clouds which can be used for further processing. Even with very dense and complex plant morphologies, such as with this cannabis plant, the method has the significant advantage of encountering minimal issues due to occlusion, allowing the plant to be viewed very thoroughly down to its inner structures. Over the past year, we have worked with our EZRT colleagues to establish a high-throughput phenotyping infrastructure at our Merkendorf location, capable of cultivating up to 400 plants in 3 climate-controlled chambers. Plants can be automatically transported from the chambers to one of 3 sensor cabins for 2D, 3D, and X-ray CT measurements. Within the climate chambers, we control environmental parameters such as temperature, humidity, light spectrum and intensity, irrigation, fertilization, and CO2 levels. This allows us to create optimal growing conditions for various plant species and conduct experiments with diverse treatment options. In our 3D measurement cabin, we plan to integrate our 3D nerve method to phenotype the above-ground plant structure. Thanks to the neighboring department, we also have access to an X-ray CT scanner, enabling us to image both the above-ground and below-ground structure of plants. This comprehensive approach provides synergistic insights into the root and shoot systems. The infrastructure will be accessible to both industry and academia, fostering collaboration across sectors, and is expected to be operational in the second quarter of 2025. We believe that high-throughput 3D plant phenotyping is a key technology for sustainable crop production. Through the NERV approach, we can generate complete and colored 3D point clouds that can be used for trait extraction. The point cloud can be further segmented into its individual components, such as leaves, and phenotypic characteristics can be automatically extracted after modeling these components. This allows us to extract relevant information from large amounts of raw data for researchers and breeders, leading to better understanding of the plant and its processes. Automated phenotyping is key to developing new plant varieties, which is crucial for feeding the growing world population in times of climate change. And with this, I would like to thank you for your attention. We warmly invite you to connect with us at Fraunhofer EasyRT.